I'd like to welcome all of you to our last session, our afternoon section of our workshop. And we have been hearing a lot about microscopy, and to stay in the bioimaging field, but now not using microscopy, but using other tools. We are going to have now a talk by Dr. Adriana Tavares, who is the head of the Preclinical PET facility from the University of Edinburgh, UK. And so it's going to be, I am sure it's going to be a very exciting section, and I'm looking forward to Adriana's talk. And I'd like to thank you, Adriana, for accepting our invitation to talk in our very first workshop from Bioimaging Net. So Dr. Adriana Tavares graduated from the School of Health Technology of Porto in Portugal with a nuclear medicine Bachelor in Science with honors degree. And she has worked for over 10 years in clinical and preclinical radionuclide imaging and obtained a master's degree in biomedical engineering from the Faculty of Engineering of the University of Porto, Portugal, as well as a PhD degree in radio tracer development from the Faculty of Medicine of the University of Glasgow, UK. Post-completion of her PhD, Adriana took a position as image processing analyst and later imaging consultant at the Molecular Neuroimaging MNI in New Haven, Connecticut, in the United States. Her research is focused on a novel selective PET and SPECT radio tracers and repurpose of currently available radio tracers as imaging biomarkers for different clinical applications. She serves as a reviewer of various journals, funding bodies, and international congresses, and she's the co-chair of the Standard Group of the European Society of Molecular Imaging, and she's the founder of PET is Wonderful Group, a very interesting initiative, I would say. And if you want, Adriana, to share a link in our chat for people from our audience to know about this very interesting initiative to, I think, to make PET more affordable and accessible. Nice. So anyone who is interested in PET should just type in our chat the link. And finally, she's the member of the Molecular Imaging Committee of the Scottish Imaging Network, a platform for scientific excellence. So Greg and I met Adriana in May in our first in-person CZI meeting in San Francisco. And it's one of those things that it's sometimes the workshops, the meetings that we go are very good for the science, but are even better for networking. And Adriana was one of those people that we met in this CZI meeting, and we had a good chat. And she's a CZI scientist herself. And when we were organizing the program of this workshop, she was one of the first names that came out to our minds to talk about different methodology other than microscopy, which is powerful for bioimaging. So thank you very much, Adriana, for taking your time to be here with us. I know you were in a previous meeting these days, so your schedule, your agenda must be full, and we really appreciate you taking your time to be here with us today. Thanks a lot.
Thank you very much, Christina, for a really nice introduction. It's a pleasure to be here, and thank you for the invitation and to the organizers for allowing me to talk to you today about PET imaging, which is a great passion of mine. I'm going to try and share my screen, see if that goes well. Okay. Hopefully you can all see that.
Yes.
Great.
Perfect. It's perfect.
Perfect. Okay, so I'm Portuguese, and I think I'm speaking to an audience that also speaks predominantly Portuguese. Boa tarde e obrigado pelo convite. And so now in English, so everyone understands me, I say good afternoon and thank you for the invitation. Okay, so I thought I was going to talk to an audience that was predominantly microscopists, so it would probably be useful to start by explaining a little bit about what PET is. So this is the outline for my presentation today. So I'm going to start by telling you a little bit about positron emission tomography, or PET, and that really is a nuclear medicine technique. And as Christina just said, my first degree was nuclear medicine. So we'll start there with the basics and then move on to talk about the principles and the key applications of PET imaging. We'll then talk about how PET has really helped us to understand how the brain works. We don't understand everything about it yet, but we've learned a lot by using PET imaging techniques to understand brain function. And the final point for today's presentation is emerging systems biology and how we can learn from the very recent development in PET, which is called total body PET imaging. So I'll talk about that later on today. So what is nuclear medicine? So nuclear medicine is a specialized area of radiology that uses a very small amount of radioactive materials, also known as radiopharmaceuticals or radiotracers, to study an organ function or structure. And so really the way it works is represented in this diagram here. So we start by having a cyclotron, which is this instrument you can see. Hopefully you can see my mouse moving. So this is a cyclotron image. With that, we can produce isotopes, radioactive isotopes, that we administer to patients or small animals or any other research subjects. We also need to have a scanner. So this is how our instruments look like. So they're a lot bigger than a microscope, but in essence, the idea there is to detect radioactivity, and this is what we do with them. And the two combined produces these beautiful images you can see here of molecular processes at the cellular level inside a human body. So really, nuclear medicine, I think the reason I like it so much is because it's a combination of different disciplines. So in nuclear medicine, we have chemists, we have physicists that looked at nuclear decay. We look at maths. There's a lot of maths in nuclear medicine, and in particular in PET imaging. Computer technology, computer scientists, doctors, and of course patients. So it's a very diverse area of imaging and research. And the other reason why I think nuclear medicine is so popular and has grown over the years is because it helps us to diagnose and to treat abnormalities really early in the disease process. So we're able to detect with really high sensitivity diseases in the early stages. So here's some examples of instrumentation that have been developed over the years. And I thought it would be good to show you how the very first PET device looked like, which is this image here you can see on your left-hand side. And so it was really very rudimentary. You know, we were only able to get to the images that look like this. So, you know, we have the little dots of uptake of the radiotracer. But we came a long way. So this was the very first device in 1953. And since then, we've been able to get these much more sophisticated techniques and tools you see in the middle here. So this is actually a PET-CT scanner that we have here in Edinburgh to scan human patients. And very recently, in the year of 2018, this scanner here, called the Explorer system, that allows us to do total body PET image, which you can see here, so the whole body in just a few seconds, was developed and is now available for clinical use. So PET imaging really doesn't happen unless we have a radiopharmaceutical or a radiotracer, as I said before. And one of the key advantages of PET imaging is it has very high sensitivity. So in this diagram here, you can see different medical imaging techniques like computer tomography or CT, MRI, SPECT imaging, fluorescent, bioluminescence, and I think what really strikes me as important in this figure is two things. First, the PET imaging technique has a very high sensitivity, so you can look at really, really small amounts, so very small changes in concentrations in vivo. So we're looking at picomolar to nanomolar, really molecular cellular changes inside a human. But the other advantage of PET imaging is that not only you can study humans, but you can also look at the mouse and other species in between. So it's really nice in terms of translating technology from research into the clinical setting. And all of this is accomplished by using the radiotracers, which are basically compounds of interest labeled with a radioactive isotope. And that radioactive isotope is what allows us to track that compound of interest in vivo as it goes from the bloodstream into the organs and into the targets. And fortunately for us, there are thousands of radiotracers reported thus far, which allow us to look at all sorts of different cellular processes. So here you have an example of some of the processes we are now able to investigate in vivo using PET. And not only investigate, but we can actually quantify those processes, those molecular changes in vivo. So here's some examples. We can look at surface receptors that are present on the cell membrane. We can look at glycolytic rate. In fact, FDG that you see here is one of the most extensively used radiotracers for imaging in the clinical setting. We can also look at hypoxia, can measure that in vivo. We can look at membrane sensors, protein sensors. We can target specific enzymes of interest and see how they are working, how they're functioning in vivo. We can also measure drug transport, for example, into the cells. We can look at angiogenesis and the process of new vessel formation, looking at proliferative rate, nuclear receptors, and blood flow and perfusion. So we can already do quite a lot non-invasively with use of PET tracers previously developed. So this is quite powerful, I would say, as a technique to assess these in vivo. But we can also combine, and we do that routinely, PET imaging with other types of imaging modalities. And here's just a couple of examples. So routinely in the clinical setting, and also in the preclinical setting, we can have a PET image, which you can see here, combined with, for example, a CT image, we can also see here. So this is an example of an FDG study that looks at glucose metabolism in vivo with PET-CT. We can also combine different radiotracers. So the image on the right is an example where we have brain function being assessed with different radiotracers. So we can look at things like mGluR5 receptors, and we can look at adenosine 2A receptors, we can look at cannabinoid type 1 receptors, or serotonin type 1 receptors A. So all of that can be accomplished by using the same technique, that's PET, but with different radiotracers. The other thing that has made PET very popular in the field of research has been its use as a companion biomarker for drug discovery. So what we have been able to do with PET for a few years now is to actually select or help select the patients that will benefit the most from new therapies. And so here's an example of how we've done that so far. We can see in this image here, so these are PET images collected after injection of a radiotracer that labels beta amyloid in the brain. And so what we've been able to do that with PET was to select patients with Alzheimer's disease that would be included in drug clinical trials and other patients that will be excluded from clinical trials based on their PET outcome measures. So this is really important and a very powerful tool for drug companies developing new therapies. The other way that PET has helped drug companies has been by selecting the right amount of the drug that we need to give to patients in order to have a therapeutic effect. And the way we've done that with PET was by using scanning the patients before drug administration, after drug administration, and then assess how much of the drug engages with the target of interest. And for most of pharmaceutical companies, as you can imagine, this is a really important piece of information because it will allow them to select not only the drug dose they need to give to patients in clinical trials, but importantly assess their production amounts that they need in order to make the drug available widely. And in this particular study I'm showing here, we actually found that this drug we only had to give this drug in microgram levels, not the typical milligram levels. And that is a 1,000-fold difference, which is really important if you're a pharmaceutical company trying to go into the market with a new compound. It would significantly reduce the costs as well of making that drug available. Okay, so that was a bit of a flavor of what PET imaging is and nuclear medicine technique as a nuclear medicine technique. But really, how does it work? I thought that would be important to describe for this audience. So the way PET works is we have the radiotracer, as I explained, that gets injected into the research subject, and that could be a human or a mouse or any other species in between. And from that, we have a biodistribution of the radiotracer in vivo in that research subject that we can track because the radiotracer contains a radioactive isotope that is decaying. And as that decays, we're able to detect the gamma photons that are produced through the decay process. And that is done by using this camera here, which is basically the PET camera. And those images are then reconstructed and processed, and the final result is what you can see here on the left-hand side corner. This is an image from the total body PET scanner that I mentioned before, and this is an image of a mouse that we collected before. And these are 3D images, as you can see here, which is always very, very, very handy for us to look at in detail across all the different organs. So the other thing we can do with PET, and we do that routinely, is we scan not only once, but multiple times over a time window. So in here, you can see the same animal between 0 and 60 minutes post-injection of the radiotracer, where we have mostly vascular components. So you can see the aorta, the heart, and the kidneys. And then if we scroll into the next animal, you're able to see a little bit more contrast of soft tissue. You see more in the kidneys and a clearance from the vascular pool. And then if you do that at 1 hour post-injection, all we see is the skeleton. This animal was injected with the radiotracer that binds to bones, and that's why we see the bone uptake at 60 minutes post-injection. And so if you imagine doing this multiple times, we can then plot that in a graph, which is what you can see here on the right, where we have the time that we took those measurements versus the amount of radiotracer that is in different organs. And that you can see here, for example, in pink, we have the bladder, the urinary bladder. In gray is the femur, which is one of the target tissues, and then all the other structures here. So the heart, the lungs, the kidneys, and the liver. So this is really powerful dynamic information. Not only we have 3D images, we can also see those over time. So we can look at dynamic processes in a very active, in a very quick and dynamic manner. So just to sum that up, in PET imaging, we have two types of protocols, predominantly the dynamic protocols, which are the ones I explained in the previous slide. And they are very helpful to research context, and they produce a lot of data that we need to then analyze. They are very useful for kinetic modeling, which is something I'll talk more in a minute. And they provide basically full quantification of that biological process of interest. So if you're looking at a particular receptor in the cell membrane, with dynamic protocols, you can really quantify how much of the tracer binds to that receptor, how much it's eliminated, and how much gets into the tissue. So really powerful tools to assess physiology and pathophysiology. However, they are very time-consuming, and they're very complex sometimes to set up. And I'll give you an example of our setup here in Edinburgh in the next few slides. But the key thing here is when we have a dynamic protocols, we scan the same specimen, the same subject over multiple periods of time. We then collect data on specific regions of interest. So in this case, we have the striatum and the occipital cortex in the brain. We then produce these time activity curves where we have the tissue and we have the blood curve. And from those two, we can then do what we call kinetic modeling and estimation of parameters of interest. So that's protocol one. The second protocol that is extensively used in the clinical setting in the context of oncology, for example, is the static protocol. So for that protocol, we inject the radiotracer, we wait a specific time period, and then we scan our subject after that uptake period or interval period. These are a lot simpler to implement and to do, which is why they are so extensively used in the clinical setting, but they are a lot more limited in terms of how much information we can extract from them. And so here's an example of our setup here in Edinburgh. So we do a lot of small animal imaging, rats and mice. And in this setup, you can see what I was trying to explain before. It is a very complex experimental setup when we want to do dynamic PET studies for kinetic modeling, because for that we need quite a lot of different instrumentation that you can see in these images. So I need to have a pump here which circulates the blood from the arterial side through the venous side in the small animal. We need to have a PET scanner. We need to have a detector that measures the amount of radioactivity in the blood of the animals. We need to have an acquisition station. So here you can see the curves being formed over time. We need to be able to monitor the animal throughout the measurement. And for that, we have a rectal probe that controls the animal temperature. We also have a respiration probe because we are required to check animal vital signs throughout all of the measurements. And we have an arterial venous shunt, which you can see here, which was surgically induced in the case of the animals. And therefore, this really is a complicated setup, as you can see. And all of that would produce these kind of curves you can see up here, where we have the blood curve and then the tissue curve. The blood curve is measured by this instrument here and the periphery of the animal, and the tissue curve is measured by the PET scanner. So when we have that, we then do a lot of maths, and don't worry, I'm not going to talk a lot about maths, but just to give you a bit of an overview of what you can measure with PET. So we inject the radiotracer into a blood vessel, into a human or an animal. And from there, the radiotracer gets into the tissue, gets into the organ of interest. And this is where you can see represented by these squares in blue, yellow, and green. And that is what we call K1. So this value here, K1, is a proxy of perfusion. So we know how much perfusion is going on in that tissue. And then from that, we can also calculate how much of what's in the tissue is binding to the target of interest. So for example, if you're looking at a receptor in the membrane of the cell, that is your target of interest. And we can measure how much of our compound, our radiotracer, binds to that particular target. And so this is very classic pharmacology. So if you remember pharmacology from your early years in research, you would remember the law of mass action. And that really is what we're measuring with PET. We're measuring this binding to the receptor and the coming off of the receptor, which we call in the kinetic modeling as K3 and K4. Of course, this is a very complicated design that you see here. So most of the time, we're not able to measure K5 and K6 because the PET images have a lot of noise, which prevents us from doing that in a reliable manner. And so we simplify this model to something that looks a bit like this. So we measure the transfer from the blood into the tissue and back, and then within the tissue, how much of that radiotracer binds to the target. So these are the different rate constants we can measure by doing kinetic modeling with PET. So we get the blood measurements, we get the tissue measurements, and then we can calculate all of these different constants. So K1 is really the flow, the transfer of the tracer from the blood into the tissue, and the other constants, as I explained on the previous slide, are to do with the binding to the target. And if you want to make things even more complicated, we can also calculate something called volume of distribution, which is very important for pharmaceutical companies because it tells them how much of that compound is engaging with the target of interest. And that is calculated using the equations shown here. But don't worry, don't expect you to remember all of these things. It's just to show you how much information, how much data you can get from PET studies. And you can get even more data because you can then calculate what we call binding potential, which is a measure of receptor or target density relative to the affinity of the compound. So really is pharmacology at its best, and it's a lot of different outcomes from a single PET session. Okay, so we've covered the basic principles of PET imaging and also kinetic modeling. We talked about nuclear medicine and what that is, and the next is just more practical examples of how PET has helped us to understand brain function. And I'll give you some examples of experiments I've conducted over the past several years. And so I think we can all agree that the human brain is a very complex organ. And I always like to do this analogy of the human brain, you know, synapses firing inside the human brain, how many neurons are there, and there's billions of them. And there are also billions of stars in the Milky Way. So when we're trying to study the brain, we are really trying to study something very complex. It is the most complex part of the human body, and we know very little about it, mostly because it's not always accessible. It's really hard to study that in a non-invasive way. But fortunately for us, PET imaging is an excellent way to non-invasively assess brain function with very high sensitivity. And so that's what we're going to be talking about the next few slides, how nuclear medicine techniques, and in particular PET, has helped us to understand how the human brain works. And I always like this analogy. To me, looking at different types of brain cells with different properties and functions, is a bit like looking at different types of stars with different properties and functions. And so how we decode that with PET is really quite fascinating. I'm going to give you some examples of that. So the first example is looking at protein aggregates in amyloid, so in particular amyloid plaques in Alzheimer's disease. And so PET has been used extensively for that. And so I think you're probably all familiar with Alzheimer's disease and the consequences of that. And what we've been able to do with PET and radiotracers is to quantify how much of that amyloid protein plaque deposition is happening. And so the image you can see here on the right is an example of a healthy control, so a healthy human versus a patient with Alzheimer's disease. And you can see a significant change in the color scale. And in this case, yellow and red means more of the plaques. And you can see that, as expected, in the Alzheimer's disease patient, there's a lot more amyloid plaques compared to the healthy subjects. And the diagram in the middle is just to help in terms of terminology. So in the next few slides, I'll be showing predominantly horizontal sections of a 3D PET image, but you can of course look at sagittal and frontal and coronal in the same way you do for other techniques like microscopy, but most of the images would be in horizontal plane. So the other way that we PET and nuclear medicine imaging techniques, and in this case I'm going to give you an example of a SPECT, or a single photon emission computed tomography. It's a very long name, but it's short for SPECT, which has been used to understand how brain receptors that are responsible for our on and off responses, or the go, no-go responses, change in the context of Parkinson's disease. So when we think about Parkinson's disease, we often think about dopamine, but we not always think about adenosine, and they're both important regulators of the go and no-go responses in the brain. So here's just a schematic of the dopamine response. So in the healthy brain, we have quite a lot of dopamine. In the Parkinson's disease brain, we have less of that, so the go responses are impaired. When we look at adenosine, we have the opposite. So we have a lot more adenosine in Parkinson's patients compared to the healthy controls, which means the no-go response is enhanced. And interestingly enough, coffee is a potent compound towards the adenosine receptors. And a study we've done in a few years back has shown these lovely images you can see here, before and after coffee. So these are adenosine two-way receptors. And in this context, you can clearly see the effect of coffee in suppressing the no-go augmentation that happens in Parkinson's disease. In other words, drinking coffee makes us go further, be more active, which is kind of what we knew was happening, but this is really the proof of that knowledge using an imaging technique. So the other thing we're very passionate about is looking at inflammation in the brain. That's very topical as well. And in particular, one of the targets of interest for my group here in Edinburgh is something called TSPO, which is a translocator protein expressed in the mitochondrial membrane. And of course, we know the mitochondria is really important for chemical energy. And this particular protein has been shown to be essential to regulate homeostasis and also regulate pathological responses. And so in the context of pathological activation, there's been some studies with TSPO, but also some studies in homeostatic activation. And this graph here was some studies we conducted a few years ago for the Department of Defense in the US. This was while I was at MNI in the US, where we looked at patients with PTSD and healthy controls. And we saw that there was a significant reduction of this protein in the brains of the patients with PTSD compared to the healthy controls. But the other side of the coin is when we have pathogenic activation. So in this case, the same protein, TSPO, is upregulated in the context of Alzheimer's disease, which you can see here on this image, compared to healthy controls. So we really think this is a very, very nice target to look at not only pathological inflammation, but also homeostatic process of inflammation. And the other example I wanted to give to you today is looking at synapses in the brain. So I started this section of today's lecture about how nuclear medicine techniques help to understand the brain by making that analogy of synapses and the universe. And so it makes sense to go back to that. So synapses over the course of a lifetime is something we're very interested in Edinburgh, and we have a study ongoing to understand how they change over time during natural aging. And so the synapse is really a structure that allows the neuron to pass electrical signal from one to another. And I think you're all familiar with that concept. We have been particularly interested in looking at synaptic vesicles and specifically SV2A, so synaptic vesicle glycoprotein 2A. And there's been some new radio tracers developed in that space, and we are currently using them. But as I mentioned, the adult human brain contains a lot of neurons, so looking at billions. And one of the objectives we have here in Edinburgh is to be able to map all of these different neurons and understand the SV2A expression in all of them, which is quite difficult to do when you have billions of them. Well, it's hard to do them when we have millions, like in the mouse brain, but it's a lot harder in the human brain. So we thought we'll start the process by looking at the mouse and then take those techniques into the humans, because it will be a bit easier to scale down and then scale up as we go through the pipeline. And these are actually an imagery collected here in Edinburgh, where you can see the 3D PET data from injecting a radio tracer that targets SV2A, and you have these beautiful synapses lighting up, and we can then track that over time and see how natural aging impacts on synaptic function. And that's something we're currently working on in Edinburgh. Okay, so the final part of today's presentation is how we can then take all of that we know from PET, all of that potential, that powerful technique, and be able to, for the first time, look at not just one organ, but the whole system and how that interconnect with each other. So for that, we're going to use total body PET imaging and kinetic modeling. So we're going to combine two things to try and understand systems biology. So just a bit of a recap. PET really is that tool that will allow us to look at precision medicine and delivering precision medicine, and we do that with three layers. The first is we need to have a radio tracer, and we talked about that already. It's a bit like a contrast agent, but with a radioactive isotope. We also need to have very good instrumentation, and as I mentioned, the development of the total body PET scanner for humans is really impactful because we can now look at the whole body in just a few seconds, which is absolutely amazing. But we also need another layer, which is image analysis. So I gave several examples of kinetic modeling before and what that is and what that entails, but there's a lot more to be done in this space. So really watch this space for more updates as we progress. And so what is the total body PET scanner? So just for those that are not familiar, the images I've shown before and the scanners most departments have look like the one on the right. So we have a standard PET scanner. We can look at about 20 centimeters of any areas of the body. So we can look at the brain specifically, or we can look at the heart, but we are limited by 20 centimeters. While the total body PET scanner, which you can see on the left, allows us to see the whole body simultaneously. So we can really start studying different systems and how they interact with each other. So it's a very exciting technology, in my opinion. But like any exciting technologies, that has lots of opportunities, including we can study systems biology, we can understand and quantify drug target engagement, not only to the target, which I've shown some examples before, but also importantly, non-target. So when you think about side effects of drugs, we can now starting to quantify that as well with PET, which is very exciting. But of course, like I said, all new techniques have their challenges. And for total body PET, the main challenge is how we're going to process these very, very big data sets and how we're going to do kinetic modeling of multiple organs, which is a challenge for us in the PET field. And so I think one way we can address these limitations is by, again, scaling down to do that in the preclinical setting and then scaling up again to the clinical setting. And so just to give you a bit of an idea of how big is big, the preclinical kinetic studies, like the one I showed you, those photographs of our lab here in Edinburgh, it requires usually around 70 gigabytes of data. So it is a large file, but when you think about clinical kinetic modeling studies, we're looking at 1 terabyte per patient on the total body PET scanners, which is huge. It's quite challenging for us to handle 40,000 independent voxels of dynamic data, multi-time point data. And so the other way we can think about how preclinical can help clinical total body PET scanners is not only because they are smaller files and so easier to handle, easier to manage all of that data, but also we can, with preclinical animal models, look to validate the findings by collecting tissue, which is something we cannot do easily in humans. So validation of new kinetic models, new ways of analysis of the big data is a lot easier to do in small animals compared to humans. And finally, we would be able, because the good thing about the mouse studies, as you're probably all aware, is that we can have animals that can be genetically engineered, and we can also do a lot of pharmacological and surgical techniques that are not easy to do in humans. And so I'm going to share with you some examples of studies we've conducted in Edinburgh already with total body PET in small animals that we think will be really useful to then translate into humans. And it will be useful for multiple reasons. The first one I want to talk to you about is being able to improve the quantification and improve the kinetic modeling aspect of PET. And so the way we do that is by, again, having these dynamic collections of PET data. So again, 3D data over time, and then be able to generate these correcting factors that you can see here, which ultimately would allow us to extract from the imaging data the blood data that we currently can only collect by physically drawing blood samples. So one of the things we want to eliminate is the need for all of that instrumentation that you've seen on the slides before, and instead have just the PET scanner, just imaging data without the blood collections, and just being able to derive the same kinetic outcomes by using the image data. And being able to use the information on the vascular pools from the imaging data to do those corrections. And we think we can do that quite well. So the first example here on the left is from the imaging radio tracer that I mentioned for TSPO imaging, so the one that looks at inflammation. And the images on the right-hand side is for that synaptic tracer that I mentioned. And in both cases, we're able to use the total body images to derive not only tissue data, but also blood data, which up until now, the only way we could do that reliably was by collecting blood samples from the patients or the animals. So this is, to us, it's a major advance to the field, because we can now do 2-for-1 really from an imaging data point. So the other thing we've been developing, as I mentioned, is this TSPO inflammation, because inflammation is very important to many different processes in vivo. And in particular, this compound that you see here, this radio tracer called 18F LW223. It's a very promising compound for inflammation imaging, because it has 3 fundamental advantages compared to other radio tracers developed for the same target. The first is that this compound allows us to image the whole human population, not just a subset of them, unlike the previous compounds. So the previous compounds, the binding to the target was impacted by a genetic polymorphism that exists in humans. And that genetic polymorphism meant that only about 40% of the human population could be imaged with that particular radio tracer, which of course is not ideal because we want to be able to image the whole clinical population, not just 40% of that. And this new compound that we developed here in Edinburgh in collaboration with colleagues in Glasgow, called LW223, is able to image the whole human population. So that was really, really exciting, a very happy day in the lab. And the other reason why the compound is so promising is it allows us to detect macrophage-driven cardiac inflammation in vivo with very high sensitivity. And you can see that in here, in this image here that we call BPTC. And this red area here is the area of the infarction, so the myocardial infarction. And that red indicates a higher degree of inflammation compared to the surrounding tissue, which was very good to see. And the final reason why this compound is so promising is because recently there has been studies showing that if someone has a heart attack, they will also have an increase of inflammation in the brain. So inflammation in the heart leads to neuroinflammation. And we've been able to show that same thing with this new radio tracer, but with a much higher sensitivity. So that is also very good news because, again, we can detect disease much earlier in the pipeline before it becomes too late for treatment. But the caveats, like with everything, is when we start looking at total body analysis, we have different tissues with different kinetics, with different performance. And so here are two graphs. The first one on the left is the graphs for the heart binding of the radio tracer, and the ones on the right are the binding to the brain. And you can see the curves look different. So that means the radio tracer has a different kinetics. And to be able to quantify that in an accurate manner, we have to resort again to classic pharmacology. And so in the brain, we have with this radio tracer the typical Michaelis-Menten kinetics, which looks like this if this was an enzymatic reaction. And in the heart, we have the Morrison's kinetics, which is very different. And this is to do with the affinity of the compound to different tissues. And so long story short, the challenges we have when we do total body PET imaging is that different organs, for example, the heart and the brain, will require different kinetic models. And in fact, that's exactly what we have seen for another study. And again, this is the same data I've shown before with sodium fluoride, which is a tracer that binds to bones. And this is why you can see the skeleton over time. And this is a very simple radio tracer. It's one of the simplest you can think of. And when we zoom the time activity curves to really see the first 5 minutes of data, you can see the curves are very different from each other. And so that means for each organ, we need to have a different model to quantify that data. And so what that boils down to is the heart, the lungs, and the liver. For this radio tracer, we need to have a one tissue compartment model, which you see here on the top. And then in the middle here, in the middle row, we have what we call a two tissue model for the femur and another two tissue model for the kidneys. So this is very technically complex, as you can imagine, very labor intensive, very time consuming. Not ideal for us to try and get the most out of the total body PET data in a simple way. So in here we can see that the time that we scanned, so for example, these first 5 graphs here were only the first 3 minutes of data. And you can see the pink curves, which are the model curves, fit the measurements, which are the blue dots, really well. So that's what we would like to see. But if we scan the same animal for 60 minutes, then the pink curves here, which are the model curves, they don't feed the data as well, which then causes problems when we look at the quantification, which are the graphs on the right, where we see very big differences if we use a short time acquisition or a long time acquisition. And that's true for different constants that you can see here. So there's quite a lot of work to be done in this space of total body PET analysis. And here is another way to look at that data. So if we have a short scan, you have quite a lot of correlations between different measures that we get from kinetic modeling. And when we have a long scan, that gets worse. So we need to be mindful about how long we scan our subjects when we're doing total body PET scanning. Okay, so just the take-home message for total body PET imaging. So we have great opportunity. We can now take the total body information and be able to get blood data without having to collect blood, and that's very good. And we are very close to deliver on that opportunity. We also can assess multiple systems using a single imaging. And we are close to that, but we need to be aware that different organs will have different behaviors, which is to be expected because they have different physiology and different functions. So we are kind of getting there, but not quite there. And finally, if we want to learn more about how different organs work together, unfortunately, the tools available so far are quite limited, including things like parametric mapping, which is a way to do kinetic modeling with visualization on 3D. And so for this, we're not really there, and we need more innovation. So the final few minutes of today's presentation, I just wanted to talk to you about network analysis, which is something we developed here in Edinburgh to look at total body PET scans. So for that, we basically have all of the different organs over time, which are these time activity curves. They are then computed as correlation matrix using Pearson values. And for that, we can have these networks here. And they are very useful to explore multiple data sets and large data sets because it can be scaled quite quickly. And so here's an example of a study where we had a mouse was injected with FDG, which is a marker for glucose metabolism. We scanned that for an hour. And when we conducted the usual analysis, which is called SUV, so standardized uptake value, and then we conducted the kinetic modeling and the network analysis. So this is the images of that study. So we have a CT image, we have an FDG image, and you can then see the quantification of the different bones. So this is a study where we were really interested in looking at bone metabolism. And those different bones, by looking at the classic analysis, all we can say is that the bones in the axial skeleton versus the appendicular skeleton are different. But that's about it. But when we do network analysis, we can see clearly that the spine is the most dissimilar bone from all the other bones. And in fact, it's the only bone that the glucose metabolism significantly correlates with the density of the bone. That doesn't happen for any of the other bones, which was the first time we've been able to show that by looking at network analysis. And so in collaboration with colleagues at UC Davis, we did the exact same thing we've done with the mouse in humans using that Explorer system, and we found the exact same thing. So the spine was once again that organ that was dissimilar to the other ones, to the other bones. And interestingly enough, we saw significant differences between the mouse and human in different bones across all the skeleton, but their energy networks, according to the network metabolism, were comparable, which was very exciting to see. And how you're asking, why is this important? Well, we apply the exact same methodology in a population of lung cancer patients, using a technique we call molecular fingerprinting, which allows us to identify patients that would survive longer than others to their lung cancer. And this was before they started any therapy, before we start changing the disease course. This is really important information because it can produce a very reliable prognostic marker for cancer patients, which was very exciting to see as well. And so really the take-home message from this part of the lecture is that to study systems biology in vivo non-invasively is now possible with total body PET imaging, that we can now do multi-organ data mining in vivo and non-invasively again with total body PET. And this is the first time that we have a real opportunity to understand how the systemic effects of local diseases can influence disease courses. And that's really important, I think. And we're also able to better support drug discovery because we can look not only at target engagement, but also off-target and side effects. And so what we propose to do here in Edinburgh is to go from today's medicine of looking at specific organs, but instead of doing that in a piecemeal way, we can look at how they interconnect in these fingerprinting systems, biology, medicine, vision. And with that, I just want to thank all the funding bodies that have supported us over the years, and my team. We can see their pictures here, the PACE wonderful team that made all of this possible. And thank you for your attention.
Thank you so much, Adriana, for such an interesting data, exciting, exciting new avenues that are open for you to explore. Do you have time for a few questions?
Yes, I do. Yeah.
So there is one question here, Adriana. How nephrotoxic are the traces used in your studies?
That's a very good question. They're not like contrast agents. So I think, when you think about CT or MRI, you need to inject very large molecules, very dense contrast agents that are nephrotoxic. But in the case of PET, they are not. We have studies looking at the symmetry, so the only thing we need to be careful is radiation dose. But all of the PET traces that are injected in humans have a lower dose than, for example, a whole body CT. And so they're not toxic from the point of view of chemical toxicity, but they're also not toxic in terms of radiotoxicity.
Great. There is another question, then Greg raised his hand as well. Do you use technetium? Do you have radiological damage?
We use technetium for SPECT imaging, and that's a commonly used isotope. Most of the PET studies are done with either fluoride-18 or carbon-11, which are shorter-lived isotopes. And we don't see any radiological damage. I've done some work years ago in terms of radiobiology and how technetium might impact on cell function and others, other isotopes. But in humans, the principle is we inject such a small amount that we are at what we call tracer dose levels. So they don't have pharmacological effects, and they also don't have effects in terms of radiation damages. In order to do that, we'll have to use different isotopes that have a higher half-life, and also they emit different types of radiation, which is more damaging to the cells.
Great. Yeah, Valeria, thanks for your questions. And she says, thank you, fantastic tool and approach, which I 100% agree.
Thank you.
Greg, do you have a question? We can't hear you. Is that him?
Back again now?
Yeah, now we can hear you. Go ahead.
Okay, so the first question is, do you think the resolution will PET resolution increase using either the equipment or algorithms, or just curious about, I'm sure people are working on this, I just didn't know how close it is.
Yeah, that's a very good question. I mean, in the preclinical setting, our resolution is about 0.7 millimeter in vivo. And in the clinical setting, it's about 5 millimeter with current technology. But with total body PET technology, we can reduce that as down as 1 millimeter in humans, which is quite remarkable. And I think even more in the future, so this is what we can do now, but even more in the future, as we optimize the reconstruction algorithms and the methods of analysis, we can try and squeeze that even further into really, really high resolution. And then, of course, we have the option of doing ex vivo. So the same techniques I've shown today, we can do ex vivo in tissue sections or in specimens, in organs or other specimens with much higher resolution. So for that, we use a technique called autoradiography that can be as low as 10 micron of resolution, which is quite impressive, I would say.
Okay, the second question linked to that: are there small versions of these PET scanners, so for use in research labs? I mean, most of them are in hospitals, but I don't really know if there's a research version available.
Yes, so we have 3 of those here in Edinburgh. We have for small animal imaging, so you can scan mice and rats and small specimens, not just in the clinical setting. And there's some research at the moment to develop even smaller versions where we can look at, say, cells and in particular body-on-chip technology, which is very topical these days. So that's also been developed at the moment.
Perfect. Thank you very much. Great talk.
Thank you.
Any more questions? Adriana, when you talked about SV2A, you came close to my heart because, I study synapse transmission, and I was wondering why you chose, or people that are using SV2A, why did they choose SV2A as a marker rather than other synaptic proteins? It's because of abundance, probably.
Yeah, that's a very good question. Yes, because of abundance, but also because of compounds that were available for that target. So, the compounds that we are using as radio tracers were actually developed as an antipyretic drug, and they we need to have core chemical structures to begin the development of the radio tracers. So it was a bit of convenience because we had other compounds already out there that we could use, and also because the target is very dense, very highly expressed. But there are other markers of synaptic function that have been developed over the years, and they're not as successful as these SV2A, which is why I think the field kind of moved towards SV2A as well, because they work really well and we have really good image quality. But there are other compounds under development that can also be used for other synaptic targets.
Nice. And one curiosity, is the price of the radio tracers still an issue?
Yeah, that's a very good question. It is still an expensive technique, but what we know is that with these total body PET scanners, we can reduce that, because one of the things I did not discuss too much is that these new scanners, we can inject potentially up to 40 times less and get good image quality. So that means, one batch of radio tracer that we use for one subject now could be used for 40. So that would reduce the cost of the technique long term, which I think would be very helpful to allow as many people as possible to use this technique.
Great. Thank you.
You're welcome.
Any more questions? So, Adriana, muito obrigada. It was really nice seeing your work, getting to know you a little bit more, and I hope to see you next year, perhaps in San Francisco. We don't know. We're gonna have another in-presence meeting, but thank you so much.
Yeah, sounds great. Thank you so much for the invitation. It was a pleasure to be here, and yeah, thank you.
Thank you.
Great.